Hi, thank you for coming to see my poster. My name is Ioana Grigoras. I am a third-year DPhil student in clinical neuroscience at the University of Oxford. And the topic of my poster is the effect of baclofen, a GABA B receptor agonist, on motor sequence learning. So GABA, the most common inhibitory neurotransmitter in the human brain, is involved in the process of motor learning. Previous studies have showed that GABA levels in M1 decrease during motor learning, and decreases in GABA levels correlate significantly with better motor learning. This means that modulating GABAergic inhibition can represent a way to influence how people learn new motor tasks. This is what I set out to study in my project. How does pharmacologically modulating GABA activity affect motor learning? In order to answer this, I conducted a pharmaco-MRI study using baclofen, a GABA B receptor agonist, as a pharmacological intervention. I asked young healthy participants to come to the lab for 2 identical sessions around the week apart. The only difference is that on one of the sessions they took baclofen, and on the other session they would take placebo. After they took either the baclofen or the placebo, they would have a long MRI scan. During the MRI scan, they would perform a motor sequence task, that's the serial reaction time task, and a motor non-learning task, the action selection, that helped us acquire data about their reaction times and action selection times. We also got neuroimaging data during task performance, so task fMRI and resting state fMRI. We also collected magnetic resonance spectroscopic imaging data, which gives us measurements of neurochemicals in different regions of the brain. We saw that baclofen significantly impaired motor sequence learning. So behavioral data from the action selection task showed no difference between baclofen and placebo sessions on either the pure reaction times or the action selection times. However, on the motor sequence task, we saw a significant impairment in the performance of participants on the session when they took baclofen compared to the session they took placebo. We then compared the patterns of brain activity during task performance between the 2 sessions, and we saw that baclofen decreased movement-related activity in the thalamus, and it also increased learning-related activity in the frontal pole. We then looked at the MRSI data. So this is an example of an MRSI map of the ratio of GABA over creatine in one participant. And this is a color-coded map. So in each of these 5 millimeters by 5 millimeter by 15 millimeter voxels, we have a value that represents the ratio between GABA and creatine in both motor and premotor cortices. When we analyzed the MRSI data, we saw no difference between M1 GABA on baclofen and placebo sessions before motor learning, but we actually saw a significant difference between how GABA changed during motor learning. So if during the placebo session we saw this decrease that we expected from previous studies, during the baclofen session there is an increase in GABA in the motor cortex. So we analyzed this data using a 2-way repeated measures ANOVA, and we saw a significant block by treatment interaction. We then looked at reaction times during random blocks and how they correlated with pre-task GABA in the left premotor cortex and M1. And we saw a significant correlation of GABA in the left premotor cortex, but not M1 in the placebo sessions. In conclusion, we saw that baclofen impaired motor sequence learning, but not reaction times, action selection, working memory, or alertness. Neuroimaging analysis showed different patterns of activation during serial reaction time task performance after baclofen administration compared to placebo. We saw some baclofen-related motor impairments that were associated with changes in M1 GABA, and this is clinically relevant because stroke survivors receiving baclofen as a muscle relaxer could subsequently suffer from impaired motor rehabilitation. Thank you.